data within cBioPortal, and it has two different mechanisms for providing that data set. We're still working on it to make it the most user-friendly package as possible by sort of avoiding users to use the API directly and provide helper functions for the users so that queries are much easier. So if you're interested in exploring cBioPortal data, downloading it, you can check out that package. I also have curatedTCGAData is a project that I worked on right after MultiAssayExperiment that coordinates TCGA data with the help of the Waldron Lab and the curation of subtypes. We've added those subtype information to the package, and we recognize that many tools exist for downloading TCGA data, but curatedTCGAData provides an integrative representation using multi-assay experiment, and it's easier to analyze your data when all of it is coordinated, and you can create a subset of your data quite easily. So that is an experiment hub package. The user doesn't really need to know all the details about how that experiment hub works, but curatedTCGAData makes it really easy to download a data set from TCGA. And we provide about 33 different cancer types, and it constructs your multi-assay experiment on the fly. So I have a couple of reference vignettes here. So if you were browsing on the rendered website, you will see that there are a list of available studies in the website. You can also, you might be able to click on this link as well here and open up the vignette corresponding to that. So I have a couple of tables that sort of give you some description of what's included in the package. And thanks to Ludwig for providing a descriptive table of these omic types in curated TCGA data. And the other package we'll touch upon is the TCGAutils package, and it allows for easier handling of samples and metadata and separation of samples, sample types, and provide some useful operations for working with the barcodes, reshaping the TCGA data, and things of that nature. So it provides sort of helper functions in three major areas. So it can sort of convert the row annotations to genomic ranges and identify and separate samples. Say if you wanted to separate tumors versus normals, you could with the helper function within a multi-assay experiment. And we working with identifiers can be a bit of a hassle, or so we provided a number of functions to translate some TCGA barcodes and interpret those through TCGAutils. So how does all of this all sort of fit together, all of these packages? I prepared a small schematic of how that works, more or less. So you can see that TCGA is the major repository, and then we have our pipeline that the broad firehose pipeline that goes through our TCGA toolbox package, which I have recently taken up the maintainership of the package. And then I have the MultiAssayExperiment-TCGA pipeline, which takes the data, cleans it up a bit, and generates multi-assay experiment objects from that are served through the curated TCGAData package. And then we have the cBioPortalData with our package that provides interface to that data. So I'll go into a brief summary of the data classes that are important. And I'm assuming that we are pretty new to Bioconductor, so I will go over some briefly just what these data classes are about. The summarize experiment is a data class for representing expression data. And one thing to note is that the expression data across the assays has to be uniform of this, or in other words, of the same dimension. And you could optionally provide range data in the rows and create a range summarize experiment. And it has sample annotations, which we call data object here, and or component of the class, and it allows for metadata as well. The other thing that, so it's a major player in Bioconductor, the summarize experiment, and you can see that many other classes build up on it or build upon it. For example, the single cell experiment class inherits from summarize experiment and extends it. So it's a very important class if you're working in Bioconductor to get familiar with. RaggedExperiment is something was started out with the need to represent genomic ranges data as a matrix format. And when we were working on multi-assay experiment, we designed a class that tried to do that, but we formalized it in RaggedExperiment with the help of Martin Morgan to create the Ragged experiment class, which, as you can see, has a G-ranges lists in the assay and does some magic to represent the G-ranges list as a square or a rectangular matrix. So the schematic gives you an overview. When you have ragged ranges or non-matching sets of ranges across the samples, you would use a ragged experiment. If you had matching ranges, you would be better off using a ranged summarized experiment. So in those cases where you don't, the ragged experiment class is recommended. And you have some operations to reshape the representation of the matrix. And you can do a simple one by stacking, or QReduce by using a window of a region of interest window and subsetting your data that way, or matching the ranges based on their positions across all the samples. So now I get to the integrative container multi-assay experiment. And here we have a 3 components of the multi-assay experiment class. One is the experiment list, which again coordinates those different data sets with different dimensions and even different observations per patient, as you can see with these 2 green lines. So the experiment list puts that all together as a S4 type of list, and the call data has the phenotypic information embedded as part of the object. So you can subset based on some kind of feature in the patients, if you were using patients. You can easily subset with the call data structure here. And the sample map structure basically coordinates all of those, the patients and measurements and the names in the columns of the experiments. So it's sort of a graph representation of all of the data that we have in the object. So you can have ID-based assays or experiments here, or you can have range-based. So it does support things like ragged range summarized experiment or RaggedExperiment as well. And so that's an option. So you can subset by a range if you had some range objects in the multi-assay experiment. And if you want to look at what classes are supported, you can click on this little triangle to see essentially the requirements for the class requirements for a for inclusion in a multi-assay experiment are pretty simple. You need to have a sort of a subsetting operation, a bracket subsetting operation, and some dim names. So that can support many number of classes here, like matrix, a summarized experiment, etc. So you can have a look at that list if you like. And then lastly, the matched assay experiment class. Here is an extension of multi-assay experiment where all the samples and patients are coordinated, so they have one sample in each assay. And you can coerce to matched assay experiment from a multi-assay experiment using these two functions. So that's the overview, and we'll get it into the interactive part of the workshop in a few seconds. So if you had time to look at the polls on the Pathable website, there is a question there that you should be able to answer now. I'll go over the polls at the end, and I'll clarify anything that needs to be clarified. For now, we can start with our interactive portion by building a multi-assay experiment from scratch. First, we'll look at our mini ACC demo. This is a data set from TCGA that has been condensed into a few assays with a few observations, and it's part of the multi-assay experiment, so you can load that by doing data mini ACC, and then invoking the mini ACC object by highlighting and running that. I do have a Shiny demo that I want everyone to take about 10 minutes to work through. You can click on here, but it may be, so this is hosted on shinyapps.io, but I think it may be easier to host it locally and faster probably. So what we're gonna do is go to our files here. So we click on the files tab on the right-hand side, and then go to the inst folder, inst tutorials. And then we're gonna click into the exercise multiassay.rmd file. So that will open up an RMD file, and we're going to click on run document at the top. And this is a learn R Shiny tutorial. Once we click that, it should be pretty quick to give you a pop-up. Make sure that your pop-up blocker is disabled for that. So let me do that on my end here. So if you're on Chrome on the top right, you'll see that the pop-up was blocked with a little X on there. Just click on that and allow always, and then try again. All right. So I prepared this Shiny demo for instruction, and we can go through this ourselves. In this tutorial, we'll learn how to extract components of the multi-assay experiment data structure, and use the constructor function to create a multi-assay experiment from the individual pieces. So we're going to use our mini ACC example to both extract and then to reconstruct a multi-assay experiment. And what we have here is our schematic that we saw earlier to study this for a little bit and see what functions we need to extract those component pieces of the multi-assay experiment. And they're listed at the bottom in the black text here called data, sample map, experiments. So if you scroll down a little bit and click on continue, you'll be able to, you'll see the instructions here. So we're going to use the experiments function to take the experiment data out of mini ACC. So we'll type experiments. And the neat thing about RStudio and the Shiny is that you have autocomplete available. So you can see what functions are in there. And we want to type mini ACC, the object, and run the code. So if you have, so in line 2, I have the printout of multi-assay experiment. That's a little bit distracting, so I will comment that out with a hashtag and rerun the code here. I'll make this a little bit bigger. And when you run experiments on mini ACC, you'll see that we have an experiment list class object with 5 experiments in there, and you can see that the columns and the rows don't necessarily match across all the experiments, and that's one of the strengths of multi-assay experiments, is that you can have assays that don't exactly match even by samples or patients. So so that's our experiment list show method or printout. You click on continue. It gives you a bit more of a note here. It's a list class. So now we want to get the phenotypic information of the multi-assay experiment. So I'm going to comment this out again and do call data mini ACC and then run the code. So you can see what's inside a small real-world data set in what's involved in what columns you can get from a TCGA type of data set. Okay, so we get a check mark and then we go continue. So now we want to extract a column from call data. So we didn't have this in our schematic, but call data is a data frame. So we could use call data mini ACC and then get use the $sign and then the name of a column. So for example, I can go back up and look at all of my columns, say, so actually we're extracting the race variable. So we can type $race and run the code. So that's how you would extract it. But multi-assay experiment also has a shortcut that you can do mini ACC $, and you can see that all the columns in the call data are populated here in this helpful tooltip. So you can click on race, and that's another way to extract data from the call data. All right. So now we look at our assays representation. That's the experiment list. But assays is a function available in summarize experiment, which gives you all of the a matrix representation as in a bunch of matrices in a list as output. So you can use that to sort of reduce your data as matrices and use the assays function to extract that as much as possible as matrices. So so we can do a class on assays as the instructions say. And then run our code. It's a simple list containing a number of, so we'll try sapply. So we want to see what classes are in that list, and we can do assays mini ACC class just to explore a little bit of what's inside there. And you'll see that these are all have been reduced to matrices if they weren't already matrices. So assays is a good way if you want to extract your data and maybe run a number of correlations or a correlation across all of your assays. You can use the matrix representation. And if you're interested in looking at the sample map within the multi-assay experiment, you do sample map. There's the same name. The extractor function is the same name as the component here. So you can do sample map mini ACC and have a look at the sample map. So it's a data frame with a number of column names that are fixed: assay, primary, and call name, and they refer to the names in of the experiments here. And in this case we have TCGA data, so we have the barcode, the participant ID barcode, and the sample barcode. So now we test our knowledge. What function do you use to extract experiments from mini ACC? So this is a little bit tricky because all of the other functions use the same name, but for the experiment list, we have a slightly different name. Maybe your first answer or reaction is to say experiment list, but actually it's experiments. So that's how we would extract our experiment list from a multi-assay experiments with the experiments function. And experiment list is a function, but it's a constructor function for an experiment list, which you don't have to use if you're using our packages such as curatedTCGAData or cBioPortalData. There's a common mistake. So now we have the multi-assay experiment constructor function. So it has three main arguments: the experiment, the call data, and the sample map. So now that we've extracted these three pieces from our example, how do we construct a multi-assay experiment? So essentially it's multi-assay experiment, and we add a comma after these. So experiments is our, so the output of experiments mini ACC is our input to the experiments argument here. And then our call data is call data mini ACC and sample map. So we use the multi-assay experiment constructor function with these three component pieces. So if you were reading in your data, you'd create something that's a list type of object from your data and put that in the experiment constructor. And then you would read in a data frame type of object for your call data. And the sample map you can generate on your own, or you could have the use of a helper function which we will look at in a second to generate that sample map. Also, if your experiments and your call data, if your patient IDs match with the column IDs in the experiments, then you can forego the sample map input, and the constructor function will recognize that they match, and they will be mapped, and it will generate a sample map for you. So essentially, that's how we construct the multi-assay experiment, and you get a printout of that, and then you pat yourself on the back because we have our first multi-assay experiment. All right, so that is our interactive Shiny tutorial for building a multi-assay experiment. Now I'll go back to our vignette here, and I'll clear this on the left. So we provide a cheat sheet for all the functions that you can use with multi-assay experiment here. We want to have a cheat sheet that's more in the flavor of RStudio, but we haven't gotten to that. But you have a list of all the accessor functions that we looked at today: call data, experiments, assays plural and assay singular, which do slightly different things. Sample map metadata, which is represented as a list, and row names and call names. So I didn't go over row names and call names, but if you do row names mini ACC, you'll see what that gives you. It's a character list representation of your IDs in the experiments. And then we have a group of functions that we'll get into a bit if time permits for subsetting a multi-assay experiment, but you can see an overview of that. We did touch upon the $ extractor function to get a call data column from our multi-assay experiment. Okay, so we went through the construction part of a multi-assay experiment. I want to point your or direct your attention to the C function for concatenating a multi-assay experiment. So for example, if you already have a multi-assay experiment from curated TCGA data and you wanted to, say, add another experiment to your data, you could by using that C function, using mini ACC and say maybe the log transformation of one of the data sets in there. And you could provide this map from argument, which tells the package to take the annotations or the sample map structure from the first assay in mini ACC and map it using those, so that it will handle the sample map rearrangement on its own, so you don't have to. And it gives you a warning because it makes assumptions that the column order in both assays are matching. Okay, so we went over the call data and the experiment list and how to extract those. I'm going to skip over that because we're running a little bit short on time. I wanted to introduce some examples using curated TCGA data. And you can see our interface to the package is pretty minimal. We use the curated TCGA data function and the TCGA cancer ID code here to get a list of all of the data sets available in that. And you can get more information if you refer to the tables that I mentioned earlier about what these annotations mean in TCGA here, if we go to that table. So let me quickly show you that table. So I have a rendered version of the website here, and you can go into the reference tab up here on the right and click on curated TCGA data reference. And you can see that here's a list of all of the available data sets that we have in curated TCGA data. So you can select one and download the data set from Experiment Hub with that code. And then we have the descriptive, sort of a small description of what each of these data sets contain. So that's curated TCGA data. For cBioPortalData, we have two main functions that come into play here. cBioData Pack, which allows you to download the sort of packaged data sets from cBioPortal as tar-gz files and represent those in multi-assay as a multi-assay experiment. I must note that we have about 70% of those all of the studies in cBioPortal successfully imported. So for those remaining 30, you might have to download them and clean them up a little bit. Or you can open up an issue on cBioPortalData, the GitHub page, and have us look at that. The data comes from the cBiogenomics portal, and they have curation efforts on their end, and then also a repository that I can point to you later if you're interested, of where the data lives and how it gets curated and fixed for any issues that may be related to the importing as a multi-assay experiment. So when you're working with data, it's always an issue, as many of you know, that data is never clean unless it's an example data set. It's never clean, so there are some curation issues that prevent us from fully supporting all 100% of the data sets provided by cBioPortal. But the other thing you can do is to download the data set and have that as a tar-gz file on your computers. So the other function is the cBioPortalData function, which allows you to obtain data from the API. And this is more of a, I guess, advanced way of getting the data, but we make it simple by providing a number of parameters. And if you provide a number of parameters, you're able to get the data quite easily. So if I have some time, I will show you how to do that here. So for the cBioData Pack function, you can explore what studies are available by doing data studies table. And you can see that these are working with cancer study IDs. So if you're interested in, say, for example, if you're at Carrie's talk, you might be interested in the breast cancer data set. So I will try to do some live coding to show you how you would do that. So first we would do library, cBioPortalData, get our studies table, and then we're going to grep for BRCA, the code. And in our studies table, there is a column that lists all of the cancer study IDs. So we're essentially looking for any studies that have that BRCA tag in them. So we can see that the Carrie study is the BRCA MBC project, WAGGLE 2017. So we can download the study using this download study function. Currently, this data set's not working as a we can't represent it as a multi-assay experiment currently. There are some issues with the data, but that's where we can step in and have a look at what's preventing the data set from being loaded as a multi-assay experiment. But in the meantime, you can download it using this download study function and the ID. So it'll ask you if you want to create a cache, yes, and then it will download it pretty quickly because it's in the cloud. And I think this is on AWS, so it downloaded super fast. So you have that tarball available to you. Now you can go to that location. You can do like untar, if I spell it right, untar and then list files. List equals true. And it's a little bit noisy, but it will show you all the stuff that's inside that tarball. So what cBioData is trying to do is to sort of automatically figure out what is data and what's metadata, and load that as a data set and represent it as a multi-assay experiment. So it's doing a lot of stuff in the background, and sometimes it doesn't all work. So that's what's available in there. Well, at least you have the data set to play around with and explore available. For those that do work, you can see what it looks like. It's as simple as entering the ID and the cBioData function, and you can see what kind of data sets are in there, mostly summarized experiment data sets and some RaggedExperiment data. For cBioPortalData, it's a slightly different workflow. We first create a cBioPortal instance object here with this cBioPortal function, and that gives us a representation of the API using some software in the background to represent the API with this with the help of Martin and the Anvil package and the and a number of other packages that we depend on. So you can use this function getStudies to do a similar operation as for cBioData for the data pack function, and you'll get a list of studies as a tibble, which you can sort through. The important column is the study ID column, which will give you what study ID to input in the cBioPortal data function. And then when we run this function, currently we use the cBio object that we use to query the API. And then we use a study ID argument with the relevant study ID obtained from this table, and then a gene panel from the number of gene panels available. So you can do this get gene panel function, and sorry, it's gene panels. So if you go to gene panels and then enter cBio, which is our API object, you'll see that there are a number of gene panels available for use as part of this function. So you can, currently it only supports gene panels, but we are expanding that to include a list of genes that are of interest to you as a researcher. But currently we have this only about 47 gene panels supported for this cBioPortalData function. So when you do that, it does a bunch of stuff in the background and returns a multi-assay experiment to you as a summarize or range summarize experiment with those copy number alterations and mutation data represented. All right, and lastly, I'll quickly go over what TCGAUtils provides. a number of functions to, again, work with the rows data. So it only works for TCGA data. It's built specifically for TCGA data, and it has micro RNA to ranges. So you can convert those to ranges, and it looks up the symbols and converts them to ranges as well. And then if you have ragged experiments, as we had in our previous example, if you have a ragged experiment in a multi-assay, it will convert those to a range summarized experiment with an appropriate sort of window for you to summarize over. Okay, so I'm going to skip those examples and not get into those. So there are a number of helper functions in TCGAutils to sort of see what samples are available in your TCGA data. So you can do sample tables and see a printout for all of the experiments in that TCGA data set. So 01 stands for tumors, and we also have a sample types table to give you help you make sense of that output here. So you have 01 is a primary solid tumor. We have 11s and 10s, which are normals down below here. And split assays is sort of a recent development and based on requests from researchers who are analyzing this data. And it, as I said,
Marcel.
Yeah.
Marcel, sorry for interrupting you. You have a couple of minutes only left.
Okay, thank you. Yeah,
Thank
I'll be
you.
quick. So this split assays is pretty neat in that it allows you to separate tumors and normals, and it will return a multi-assay experiment with two different assays in that based on their sample annotation. So I wanted to highlight those and show you that there is functionality for converting barcodes to universal IDs, etc. And the other thing I didn't get to, I was talking for too long, I think, is the subsetting mechanisms in multi-assay experiment, which let you subset on the rows, the columns, or the assays based on the position here. So I, J, or K. And a more, and I think I'll leave it at that and have you explore it on your own when you have time. And I'll take any questions if time permits. Is the R code for the Shiny app accessible? Yes, it's, the first thing in the Shiny app is the actual source code. So let me go back to this. You can see the source code here at our Waldron Lab multi-assay workshop link here. Yeah, so the source code is this RMD file. Other questions? And if you are interested, you can quiz yourself with these polls in our Pathable website. Thank you everyone for attending, and I'd like to acknowledge Levi Waldron, Martin Morgan, Ludwig Geislinger for all their amazing support.
Thank you, Marcel. That's a great tutorial. It's like come so far over the last couple of years of giving it. It's so nice and simple. And I think you also created a bunch of new fans of Learner.
Yeah, with this 1 hour format, really had to focus on what's really important
Let's see, there was another question. Could you provide the link to the PowerPoint on Tiny? I'm not sure.
Yeah, it's tinyurl/multiassayworkshop, tinyurl.com/multiassayworkshop, and I can post that.
And by the way, your Google Slides were not visible to the world. I heard a complaint about that.
Sorry about that. Should be available now. And I will post this link on the chat, in the chat here. Oh, thanks.
So we'll see you all on the poster session next. I'm looking forward to seeing how that goes.